I just saw a bit
Is there a
Spitani,
power?
Hyderabad in the opening slide. Is that new news for us?
Yeah,
Okay, wow.
I thought everybody knew.
I didn't know.
I knew.
Okay.
What happened?
Huge delay.
My goodness, what is happening?
Like a minimize really. Exactly. That's it.
Okay, fine.
Like that.
Which one will go to YouTube? That camera or
This camera. This camera. This camera.
this camera? তাহলে
Yes.
তো আমার পয়েন্টারটা কোনো কাজে লাগবে না.
No.
This will not go to YouTube.
No, that will not go to YouTube. But if you
তাহলে আমাকে
use
এখান থেকে
this,
করতে হবে।
if you use this, then it's okay. I will remove the chair.
থাক।
Would you like to use the chair to deliver the colloquium?
না না।
Okay. No, I'm just asking. If you have to move the
তাহলে I have to use my marker.
Oh, you can write on it? Oh, yes.
Right.
Oh, yes, you can. Interesting. This card.
Mutant cilia, which is
That is ভিডিওটা কোথায় আছে? ভিডিওটা
Now you can sketch it.
পিন কর, ভিডিওটাকে পিন কর, জাস্ট পিন দ্য ভিডিও।
আমি কি এক কাপ চা নিয়ে আসবো?
হ্যাঁ, চা কোথায় পাবে?
ও ক্যান্টিন বন্ধ।
ওটা কি কর না, ওটা কি কর, হ্যাঁ এটাকে পিন কর। না না না, এইটা, that's it. Okay,
এটা মেন ইয়েতে চলে এসেছে।
ঠিক আছে, হয়ে গেছে? Now check in your YouTube link. It should be pinned.
Okay, আমি একটা চা।
Thank you also. Sorry.
Till then, I think we cheers. Some of these cheers.
It's best we come forward because I'm not sure how many people will occupy this. Yeah, something. Thanks. हाँ? क्या हुआ? Thank you for coming. It was a unfortunate.
I'm glad I double checked.
Yeah.
It's always been geography holiday here in Maharashtra.
I have no idea.
It's a TIFR holidays, but we don't put it on the calendar.
I should have checked that. I actually booked this long back, so this is the problem that holidays are.
Number of times, like, what is that for? Was that the other problem?
We had one time like that, but then we delayed it by 1 week.
You don't check these things.
Error, come forward. I don't think we will get to see you.
Should be. I've been asked.
You want it? I will stop. I actually do that. No, she's equally.
Yeah, you can.
Okay, then it's fine. Then it's fine.
But I.
It's best.
Yeah, so it's stuck.
Software use case.
Use. Even the IMCD and use. And then, yeah, start.
So good.
Working. Yeah, someone.
I don't think anybody will sit over there. It's a,
I can do it.
actually, the mic is not there for the mic. No.
What are you doing?
Loud and clear. Me?
No. Yeah, I told you. I told you.
Someone will go. Someone will go.
It's been a lot. Yeah, exactly. That's why I told you.
Please sit in front because there is no mic. Sorry. Thank you. Go ahead. Yeah. Let's not go back. मैंने चाय का बंदोबस्त किया है
Because this can. I asked him whether he remembers.
He knows that that's easy, not dodgeball. That's right.
Yeah.
क्यों बोल रहे हैं
Seriously?
Wow.
ये कहीं से भी आता है।
अगर चाय जल्दी से आ सकता था तो लंच में तो
कहीं से आता है।
I just took a break because I thought some of you will take time to come.
Thanks. Okay. Just wait for 1 minute. I can shout. I invited a few fellow TIFR. Anyway, I will start actually. It's 4:05. So welcome to this to the Wednesday colloquium on a holiday. Thank you for coming on the way, and this is a special colloquium. Professor Krishanu, who recently superannuated from the system, kindly agreed that he will recount some of the interesting stories that he had in TIFR through research and connection to people. And of course, this is always a special one. And so I would like Professor Krishanu to be introduced by a senior colleague in Department of Biological Sciences from Joubert.
Okay, I mean, it's truly a delight, a bittersweet privilege. Krishanu and I actually joined about a similar time. Okay, let me do the proper introduction. Krishanu did his PhD at TIFR, student of Veronica Rodriguez, then a short postdoc in Singapore, and then a postdoc at UCSD, and returned here as a faculty member in 1998. I joined in 99, and since he had already been at TIFR, I remember sending him a mail. I mean, this prosaic introduction hides so much. I remember sending him a mail because I'm one of those agonizingly frantic planners. Okay, so what's it like? How does ordering work? How long do chemicals take to reach? Is there anything I should take with me? Krishanu and I, oil and water. If I had to pick one word to describe him, unruffled. Unruffled. First, it took him like around 10 days to realize, this frantic person needs some kind of assuaging. And his general reply was, it will all sort out. Why don't you just get there and figure it out? Okay, and you know what? It did for both of us. So both approaches work. Perhaps one with a calmer, sort of life trajectory. Okay, so then we joined. And I should say Krishanu looked exactly the same as he did.
I hope you are here.
Actually, okay, now this is like, I'm sorry, because we had kids around the same time, and I remember, I would walk around with Abhay, Krishanu with Ahan, and Krishanu walked with Ahan, and that was before Ahan had fun. I swear, Ahan ko mushi dalo, it would have been carbon copy. But in fact, anytime Krishanu's students, when they defend their thesis, it's like the fourth generation of the fantastic tradition of Drosophila neuroscience started here, started the molecular biology unit. So Krishanu is a bearer of serious history. Veronica's advisor, obesity, was handpicked by Homi Bhabha to start the molecular biology unit. And it's that tradition that Krishanu has added a completely new direction, a creative direction to, as you're going to hear in this talk. Just to prime this talk a little bit, one day I remember in a corridor, we are running to each other, and he says, Shubham, my flies can't hear. This is not you did not expect. You suddenly, turns out cilia, mechanoreceptor, that was that story, right? I mean, just unexpected scientific discoveries in his career, which I will let you do. I'm going to fill in the other stuff that he can't admit to himself. Okay, take when we came in here. I don't know, or maybe the front two rows were here when I came in, and I was looking for the speaker, because usually there is a speaker fiddling with slides, something. Some amount of free, I mean, that is normal behavior. Krishanu's absolute, true, 100% penetrant phenotype. Well, first, is that Krishanu wasn't here.
He had done his job.
all our faculty meetings, Krishanu lets all that ruffling settle in, and guaranteed Krishanu will bring an unruffled presence in after everybody has already done. So we saw that here today too. And it works, we had all settled in, all of JD's scolding, all of that had finished, right? So that's it's a useful life approach. Krishanu steered our department actually in by simply keeping the balance through some very tumultuous times. Because I think when we joined, there were maybe 6 people in the department. It was a very, very small department, and everything had to be set up pretty much de novo. Rohit, who joined also before Krishanu, Rohit, then me, and then Vidita. We were the young crowd. This will be you down the road. Everything had to be sort of set. The coursework calendar, the microscopy, the literally all the training program, everything we had to be set up pretty much. Let's see, I had a story of doing there, but now they've just gone out very good. I will fast forward to when Krishanu became chair of the department. And again, those were very complicated times. And his sense of balance and weathering ups and downs were critically needed at those times, because I think those were, again, tumultuous times in our very tiny department. He was, in fact, a mover and shaker for the Hyderabad banks. Pretty much, I think, built the sort of foundation of the idea with his own hands. And for a while, I was sure that I was going to lose my friend and colleague and was going to disappear off to Hyderabad. And that would have been all right at CIFR2 and so on. But I think now when the building is built and many people are there, I think people don't know the early warriors who took it on. And I think Krishanu should be really recognized and applauded for that. I don't actually know where he said it, because another part of this unruffled calm is that not much information comes.
It's right there. It's Hyderabad.
It's Hyderabad. Okay. I was expecting, I was hoping, but we never know. Krishanu actually left our little family earlier than anticipated, because he took on the directorship of MBRC, which is, if our department was tumultuous and complicated, NBRC, from what I heard from Krishanu, was even more so. So it's actually quite a contribution to a country to set up your own program and then to lead a department and then a whole directorship of an institute. And now he hasn't stopped, because now at Bitspilani he will be continuing his career. So I will right now let him tell us about the science, but I do want to say that you will be missed. And
Thank you, Kumar.
funny story.
Can I start?
Yes, you can.
Thank you.
Just he's going to check if there are people in Zoom.
Oh, do I need a mic?
No, you don't. Well, we don't have one. So you have
Okay,
to come.
all right.
So everybody's.
So I hope, but I hope those who are connecting through Zoom
They are able to
will be able to.
Yeah, we can hear you, Krishanu. Yeah,
You can hear us. Okay,
loud and
great.
clear. Yeah.
All
Yeah,
right.
thanks.
Good. All right. So thank you very much, Jyotishman, for forcing me to give me the talk. And thank you, Shubha, for a mini colloquium before my colloquium. But anyway, so history apart, and let's talk about the future. And what I'm going to tell you today is something which is already happening, and you will hear much more from many other sources as you progress along in the future. So I thought it is the right moment to actually highlight what is cilia, and why we should all know about this tiny little organelle of the cell called cilia. So to start with, I'll take you back to the middle of 17th century. So this is what Leeuwenhoek first observed when he made his ocular, two crude lenses stuck together with a bamboo tube. And so it's not clear who coined the term, but he definitely watched this little feet, which he called little feet, on most of these aquatic microorganisms that he could see in a pond water. Now I'll show you how these organisms actually behave. So this was later named rotifer in Latin, which means wheel bearers. And you will soon realize why they are called wheel bearers. So this is one of those wonderful videos that you can see. So they have this little cilia, which is in the mandibles, And these are multicellular organisms, about 1 millimeter long. And they use this cilia, this wheeling motion of the cilia, to suck the food into their what we can call alimentary canal, which is not really an alimentary canal in this organism. And now I will move to another structure, which you are also quite familiar with, which is this human spot. And as you can see, it has this long tail, which is highly motile, and that makes this organism or this cell move from one place to the other. This is very critical for the life. We couldn't exist if the sperm doesn't move. And every organism makes a sperm which is motile, except C. elegans. So, and also there are many individual organisms which move with this little tail called flagella. Now, these structures which I told you, flagella and cilia, not only is required for moving things, it is also required for actually maintaining our mental activity. So what you see here is the movement of the ependydymal cilia, which is in this place of the brain called, Shubha knows better. So this is the ventricle of the brain where the cerebrospinal fluid, which supplies all the nutrient to the neurons, are present. And as you know, brain doesn't have blood vessels, so the brain needs a heart. And these little cilia, which is about 10 microns long, actually do the job. And here is a demonstration of how they actually do this job. And you will see how complicated these motions are. So this is in the mouse ventricle, and this is fluorescent dextran injected while this is being imaged. A really marvelous feat of engineering to do this. What you will notice here is that depending on where this fluorescence is injected, the flow is guided to a different directions. So this movement of the cilia in the ventricle is not just an autonomous movement. It is highly regulated and guided movement. And these movements are controlled by your brain activity. So the regions of the brain which requires more ATP and more glucose, as they are more active, for example, now your auditory cortex and visual cortex is highly active, and to some extent your frontal lobes also in some cases are highly active. So those regions of the brain would need a greater flow of cerebrospinal fluid rather than the other regions of the brain, like your motor cortex. In my case, on the other hand, the motor cortex, and in addition to the other two parts of the brain, will require higher flow of fluid because that's also is active quite a lot. I am exercising my larynx. So this is the role of this cilia inside the brain. Now, not all these ciliary structures are motile. You also see these kind of structures on all other cells in our body, and every other multicellular organisms have the same structures, except few like Drosophila. And they grow these little, tiny cilium, which are immotile. And you can flow a liquid around the cell, and you will see that the cilia is highly elastic, and they are very resilient, and they can move along with the flow. And this movement, this is the kidney cells which grow this cilia, and this movement in the kidney determines how much urea and salt should be extracted from the blood and excreted. And if this cilia fails to sense this movement, then your kidney will malfunction and you will require a dialysis. Okay, so that's the important part. Now, all this was not known until 2000. Until the beginning of this century, these little structures called primary cilium are thought to be a vestige of those motile structures I showed you. It was thought that they are useless and they just don't have any role. Now, the term primary cilium was first coined by Sorokin in 1968 in a Journal of Cell Science paper, which is now highly celebrated but never cited for next 50 years. So it was rediscovered in 2003, and since then it is one of the most highly cited papers. And he described in that paper with a very, very brilliant systematic transmission electron microscopic studies that how these little tiny structures form. Now, in addition to this primary cilium, we also have highly modified sensory cilia. And you see some examples here, the rod and, these are the photoreceptor cilia, which you see in the rod cells and cone cells you see in the retina. And they are involved in sensing the light. These are the stereocilia inside the cochlea of our ear. And these are the olfactory cilia that you see over here. This is a fish olfactory cilia, by the way.
So
Which are
this cilia in the rod and cone, do they also carry rhodopsin like?
I'm coming to it.
Okay.
Yeah, I'll come to it quickly. Yes, so they display the photoreceptor rhodopsins on them. Yeah. So this story began in 1993 with a very innocuous discovery in a chlamydomonas lab in Yale University headed by Joel Rosenbaum. And he has been, dedicated his life since 1960s to figure out how the chlamydomonas grow its flagella and how it manages to move the flagella. That was his lifetime work. And when they are looking at this flagella, chlamydomonas flagella, and one of his students, Kozminski, for some reason mounted the flagella very tightly. And the flagella could not move because the chlammy was squished between the cover slips. And when he looked at the movement using the state-of-the-art video-enhanced DIC contrast microscopy, in that time it was this, this best imaging device that you can make. He found this movement, back and forth movement. So this movement was named intraflagellar transport, and it was turned out that these are these protein-rich particles which keep moving back and forth inside the flagella, which has this 9+2 microtubule arrangement that you see if you cut a cross section across this flagella. So after a lot of study within 2-3 years, it became clear that these protein complexes that move inside the flagella has multiple different polypeptides, and these are called intraflagellar transport proteins, or IFT particles for short. And still then people didn't really realize what is the importance of this discovery. It was still, although, many of these papers were published in PNAs, but that could be because, Joel was one of those editors of PNAs, and, he managed to push all these papers into PNAs. But around that time, another two very innocuous discoveries came into light. There was this mice called TG737. It was a spontaneously mutant mice in the collection of Jackson Laboratories. And this mice had a, human-like disorder. It's called Polycystic Kidney Disease. And this happens to many humans, this Polycystic Kidney Disease. It's quite prevalent. Almost 5% of human population suffers from this disorder. And this causes multiple cysts to grow inside kidney and kidney malfunctions. And it turned out that this single point mutation in this gene called TG737 actually causes this polycystic kidney disease. And then, as soon as those IFT particles were sequenced, proteins were recognized, it turned out that one of these IFT particle, complex protein, which is IFT 88 kilodalton, is actually the same gene as TG737 of mice. Okay. So people started thinking, so what's the connection between chlamydomonas flagella and movement inside chlamydomonas flagella and a kidney disorder, right? It's very difficult to connect. Still, people were skeptic, and then came this paper around the same time, where the Nobutaka Hirokawa's lab in Japan, they were studying a kinesin-like motor protein called KIF3B. And they found out that if you knock out KIF3B totally, then the mice stops developing after blastula stage. They don't form the gastrula stage. And the reason the mice stop developing at the gastrula stage is because the visceral asymmetry is messed up. And that was the first time it was realized that a cilia grown on a bunch of cells called nodal cells at the end of the gastrulating, at one of the end of the anterior end of the gastrulating embryo, is responsible for this left-right asymmetry, for which the heart goes on the left side and the liver is on the right side. And loss of KIF3B eliminated the cilia, and that eliminated the visceral asymmetry. And then finally, it turned out that the primary cilium is very critical for a particular kind of chemical signaling in the cell called Hedgehog signaling. And at that point, it was already clear that Hedgehog signaling is the key, contributor for several cancers, including glioma and several other type of cancers. And that is because the Hedgehog signaling maintains the cancer stem cell, or the hyper Hedgehog signaling. So therefore, and many other things, for example, having multiple digits like polydactyly, or having cetus like the left-right mixture. So then, once it was linked to cancer, everybody jumped onto primary cilia. And it became the most celebrated word in the research community. So since then, every issue of Nature Cell Science and Cell and many other top-rated journals, you will find a paper which talks about primary cilia. So now let's find out why is this so important. Okay. So a whole field of research, clinical research developed since then, because the discovery of the primary cilium and the proteins that make the primary cilia. And you see this whole list of names. This is not to scare you, just to tell you that several congenital monogenic inherited syndromes, which means that a single mutation, either autosomal, dominant, or recessive, can cause this kind of disorders. And this includes like polydactyly, retinopathy, polycystic kidney disease, and so on. Okay, and mental disorders. And this is because these mutations, which are connecting to these syndromes, all were found in proteins that make the cilia. And that is why these are all collectively known as ciliopathy. So now, we know that every organ in our body is affected if there is a dysfunctional cilia. And this is the kind of, chart that you see in our body, which illustrates how important it is to understand what is primary cilia. So the question is why? Why the primary cilium is so important for our physiology, development, and not only just humans, for every multicellular organism. Thank you. So to illustrate that, I divided the function of cilia into these 4 broad categories. There are many more, but this is just the gross categorization of cilia function. So from the sensory perception, which requires sensing light, odor, touch, everything involves a sensory cilia. For locomotion, we have already demonstrated that this movement actually propels the movement of the cell or the fluid around the tissue. Now, this is the most interesting part. So as you see, this is again a very partial list I prepared about 2 years back. And if I start adding to this, then it will go away. Things will spread out of this canvas. So almost every signaling that we know happens to happen through the primary cilia, right? And also cell cycle regulation. So when the cell is quiescent, like in general, most of our organ tissue cells are quiescent, they don't divide. So those cells express as a primary cilia. If the cell has to divide, the primary cilium has to be resolved, and then the cell will go into cell cycle. And this connection is very tight. And the reason this connection is very tight is because the mechanism that makes the primary cilium is the mechanism that prevents cell cycle. And that is the reason why cilia became such an important organelle for controlling cancer. Because the cancer cells have this mechanism disconnected. And the reason the cancer cells have this mechanism disconnected is because the primary seed that makes the cilia, called basal body, is actually sort of a derivative of another structure called centriole, which is essential for dividing the cells. So if the basal body transitions to centriole, the cell divide. If the centriole transitions to basal body, the cells stop dividing. And that happens through this set of enzymes called Aurora A kinase and polo-like kinase. We'll come back to that some other time. But today I'll talk mostly about the cell signaling, because which will relate to what I'll tell you what we did in our lab. So for cell signaling, I'll give you a quick primer of how the cell signaling works. It's a domino process. So typically the cell will have a receptor, and this is sort of I have drawn in the context of a cilium, which will kind of have an internal, internal connection with another set of proteins. And at this stage, when this protein, which is bound with a molecule called GDP, is inactive, okay? And there will be something outside the cell called a ligand, which is another polypeptide or a small chemical or something else, which can come and bind to this receptor. When it binds to this receptor, it starts the chain reaction, okay? And that chain reaction will dissociate this complex internally and will bind to different sets of proteins, generating different kind of molecules, like inositol triphosphate and cyclic AMP, okay? And this is what we call signal transduction. So the signal from outside the cell goes inside the cell through this chemical conversion. And after this, another set of chain reactions sets up. Now, if this kind of receptors are present in the cilium, then we will generate different kind of behavior depending on which receptor and what cell type it is displayed on. For example, in case of a neuron inside our brain, this receptor called MC4R is responsible for controlling the feeding. Now, if a hormone called MSH, which is involved in our wake-sleep cycle regulation, binds to this receptor, not in all cells, but only in this particular set of neurons, then we will stop eating, okay? If not, then we will continue eating irrespective of how much we ate. And that causes obesity, okay? And there are drugs which can control this behavior. The same cilium, if it displays a different kind of receptor, and these are all the same G protein linked receptors, then the cell will it will drive the fat cell to grow more fatter. Okay? And in the insulin secreting cells of the pancreas, again, the same kind, a different kind of receptor with the same mechanism will drive the insulin secretions upon glucose stimulation. Okay? So this is how the cilium manages to perform multiple functions by displaying different sets of receptors on different type of tissue. Now, what will happen if a different receptor which is antagonistic to this process is also expressed in the cilium? I'll tell you that story next. Okay. So here we'll talk about two different receptors, one which promotes the positive signaling and one which promotes the negative signaling. And to understand that, I have to give you the context of how the feeding behavior is regulated. And this is true not only for mice, it's also true for humans. So in case of mice and humans, when you have eaten enough and your fat cells grown fatter, it releases a chemical called leptin, a polypeptide. And that will bind to a set of receptors on a set of neurons. And there are two sets of neurons. So one, a neuron which when the leptin binds to it, will release alpha MSH, which will stimulate the MC4R and stop the feeding. But if it binds to the another set of neurons, the NPY or AGNP secreting neurons, then it will release the opposite, have an opposite effect. It will release AGNP and inhibit the MC4R activity, and you will continue eating. So typically, this feeding and non-feeding cycles are controlled by carefully maintaining the leptin levels in the blood and by controlling these two polypeptide secretions onto this into this particular region of the brain. It doesn't matter what it calls, but it's a very important region of the brain, which makes you feel hungry or not hungry. All right. But there is another receptor called GPR88, which can also, get displayed on the same neuron and same cilium. And if it is displayed in the same neuron into the same cilium, then it will prevent the MC4R to signal, and you will continue eating even though your tummy is bloated. So this is the way the two receptors can actually control and maintain our signaling inside the brain, and that can control our behavior. And this behavior is very important for our survival, right? For example, if you understand a crisis coming in the coming years where food will be scarce, we will tend to overeat and add up more fat and then, you will serve you for the next, several days or weeks without food. And that happens for animals that hibernate during the winters. And those animals, they actually have a mechanism to antagonize MC4R so that, they can eat more and accumulate more fat, okay, at a certain phase of life. So fine. So that's a different story of, for some other day. So today I'll focus on this one particular questions. So in this case, how does the neuron decide which receptor to display and when? Okay, and that is the question that I will deal with in the next several slides and with the data that we have gathered in our lab. But before I could show you my data, I have to give you another long background. Okay, not too long, maybe 10 minutes background. So for that, we first understand how the cilia looks. Yeah. So this is the electron microscope image of a primary cilium. And what you see here is what is called the basal body or transition zone of the basal body. And this is the cilium, and the green thing is the axoneme, the microtubule, cytoskeleton that maintains the structure of the cilium. What you notice here is that the membrane, which is just tightly opposed to this cytoskeletal structure, has very little space between the membrane and the cytoskeleton. So cilia is very, very tight. There is no space inside the cilium. And remember this message because that will come handy when I show my data. Now, these kind of structures are found not only in primary cilium, they're also found in all sensory cilium, which Jyotishman was eager to learn. So what you see is in the photoreceptor cilium, in the stereocilium, the kinocilium, and in the case of the olfactory cilium, they all have the same basal body transition zone structure and axonemal structure, which either contains this 9 doublets microtubule or it contains this 9 triplet microtubule. Okay, towards the base it is triplet, and then as you progress towards the tip it becomes doublet, and finally there are single microtubules like that. Okay, so this is like going in and coming out from a very tall building. So this whole ciliary structure has to build itself base up, and it also has to maintain this traffic in and out constantly to maintain the receptor flow in and flow out with this very little space available for that purpose. And because of this very little space available inside the cilium, the simple diffusion process doesn't work. So nothing actually diffuses inside the cilium, because if it does, we will all die immediately. Okay, so because it will be so indiscriminate that the cell will be thoroughly confused about which signaling to respond to. So that is why the cilia needs a very effective gatekeeper. So this is the standard process by which things move inside the cilium. The proteins are processed through this structure called Golgi, and then they are transported to the base of the cilium, and then they are kind of moved into the cilium actively by pulling through the membrane. And this active movement is carried out by this motor protein called kinesin 2. Okay. And so therefore the cilia needs a very efficient gatekeeper to decide what kind of protein to allow at what time. And the gate looks something like this. It's a very, very tight structure which has lots of polymers of proteins, organized in a crystalline lattice. And you might wonder that how anything can get past this, tight gate. And that's the curiosity which drives all of us to understand how cilia makes itself. Yeah.
What is the length scale of those structures?
So the across is 200 nanometer, and the length will be something like 100 to 200 nanometer. The transition zone varies from cilia to cilia. It can be as small as 50 nanometer.
The pore is there in the middle?
There is no pore.
No pore.
There is no
Everything is
pore.
blocked.
Yes. When you look at it then in the cryo-EM, you don't see any gap. So it's really strange. We still haven't got any idea how things move through this. So it must move when it must dissolve, conditionally to allow things to move through it. That's the hypothesis. Okay. Now I'll tell you in the context of the receptors. So in case of cilium, we already know more than 100 different receptors are displayed. But the problem is that the cilium, generally primary cilium is very small, up to 0.5 micron in length. In some cases, it can be very long. The sensory cilium are 10 microns, but generally most primary cilium are, half a micron to a micron long.
I have one more question.
Yes,
Sorry.
go ahead.
If you cut it,
Huh.
it will regrow, right? In certain cases,
You can pull out this primary cilium. It has not been done for the regular cells, but in case of chlamydomonas, you can give them a shock, and they will shed the flagella,
flagella,
and then they will grow it
regrow,
again.
right?
They
So
will
when
grow
it's
it again.
growing,
Yeah,
there is still movement of material.
exactly. So through IFT. So IFT is needed for everything, to make the cilium as well as to maintain the cilium. Okay. So that's the kind of conveyor belt which runs constantly. And this is the marvel of this molecular self-assembly. So you're looking at 500 different kind of proteins which just assemble each other at a particular order, all coming from the base to the tip. Okay. And this is not happening in solution. This is happening in a very tightly congested space where both the membrane and the structure grows together in step.
What is the half-life of any receptor that you're talking
That's a very good question, but that varies again the receptor that we are
about?
talking about. Well, for a smoothen receptor, it is about 190 hours. So depending on the receptor, for the PDGF, it is conditional. So if you have a tissue injury, then the PDGF turnover gets increased by 10 minutes to 10 minutes. Otherwise, it's very slow. It doesn't turn over at all. And PDGF is required for closing the wound. So and migrating the epithelial cells. Huh?
Why should it be exponential?
We really don't know the answers. These are the things which is beyond our reach as of now. Because we can't simulate it. And also we can't change the turnover rate. We don't have the understanding of the mechanism, so we can't manipulate it. So I'll give you an idea towards the end of this talk, which will give you an idea how it can be controlled. Yeah, so therefore the transition zone must be able to have a very, very intelligent brain. It has to recognize the receptor at a particular time and allow its entry, and it has to prevent the entry of the receptor at some other time. So how does the cilia manages to do it? Now we have some idea about it. We now know that the gate has a lipid, divisions, and particularly this phospholipid called phosphoinositol phosphate. So as you see here, there are these proteins which are known to cause polycystic kidney disease, the NPHP, MKS, and so on. And these proteins bind to a lipid called PI4,5 bisphosphate. Okay. And this lipid is present only at the base of the cilium, and another lipid called PI4 phosphate is present in this region, the rest of the cilium. This lipid, partitioning of this phospholipid actually is found to be critical for, and also cholesterol is found to be critical for partitioning a very important set of receptors called, TRP, transient receptor potentials, vanilloid channel type of receptors. These are the receptors which make you feel heat and cold. These are the receptors which make you taste chemicals and so on. Okay, so and also see light. So these TRP-V receptors and the G protein coupled receptor, the GPR161, which antagonizes the TRP-V channel, their movement into the cilium requires this lipid partition. And without that, it will not get into the cilium. Similarly, another receptor called smooth end, which is very important for hedgehog signaling and actually making head different from the legs during the development, also signals through the cilium. When the hedgehog binds to this a receptor called patched, which actually is the gatekeeper and which keeps the smooth end away from the primary cilium, the whole complex moves to the base of the primary cilium. And at the base of the primary cilium, there is an enzyme called DHCR7, which dehydroxylates cholesterol and makes free cholesterol. And there are only 4 or 5 such enzymes placed at the base of the cilium. And because of that, a few, a very small region of this base of the cilium has high cholesterol concentration. And that does the trick. When this patched and smoothened complex reaches the base, cholesterol binds to both of them. But it imparts completely opposite effect. So cholesterol binding increases the mobility of smoothened, and it goes to the tip of the cilium, and it decreases the mobility of patch so that it doesn't go into the cilium. So the patch remained where it was, but the smoothened gets detached from the patch and goes up to the tip. And then the signaling starts, and the signal goes out from the cilium into the nucleus. It doesn't matter how it goes, but that's a separate story altogether.
And this,
But this process of signaling through the cilium also triggers the end of the signal, because when it goes out and it induces synthesis of an inhibitor of R7, and that inhibits DHR7 and increases the another activity of another enzyme called CYP7A, which is a P450 class enzyme. And that hydroxylates cholesterol and stops the signaling. So you see there is a clock which maintains the signaling and stop and start of the signaling by itself. All right. And same thing happens with TGF-beta, where different lipid called ceramide is involved. So now the question is, how does this receptors that come from the Golgi and reaches at the transition zone of the cilium manages to move to the tip of the cilia? We haven't discussed that yet. So now I will come to that and discuss that through some data that we have gathered in our lab. So for these, I will introduce you to Drosophila. Now, Drosophila is one of the very good systems to study primary ciliary dyskinesia or other kind of ciliary disorders, because most of Drosophila cells don't grow primary cilium. Almost all Drosophila cells doesn't have primary cilium, except the sensory neurons. And those sensory neurons make sensory cilium. So you can actually get rid of the cilium and the fly will live quite okay. Not very happy, but quite okay. And that's what the Shubha was talking about. The first thing I learned in 2003, that you can disrupt the ciliary function to make the fly deaf. But that's a very old data. So this is the data that we got in our lab.
How does the fly move its fluid around?
Fly doesn't need to.
Hemolymph.
No. The hemolymph is moved by the heart. There is no ventricle in the brain, so no cerebrospinal fluid. Other than that, it's very difficult to understand how the fly. The rest of the stuff I really don't know. Maybe they have a villi which are more motile villi which does the job. But it's kind of an evolutionary intrigue that happened at some stage. So in case of fly, this is the hearing organ as you see here. And these are the cilium inside this hearing organ, which is like a guitar string that vibrates when the sound comes. And the Drosophila needs to hear for a very important courtship ritual. The male flies, flip their wing and sing, which you can also hear when a mosquito comes near to your ear. And the female responds to that song, and that's how the courtship begins. Okay. And in cricket, it is much more, but cricket doesn't, uses the same kind of cilium to also hear. So these are called chorotonal cilium. And what we found was that a mutation in a kinesin-like protein can make this string disappear. Okay. And remove this cilium totally from this organ. And as a result, the fly survives all right, but it is unable to balance, walk, or hear anything. And this is the primary flagellum of a Drosophila sperm. And you can see that there is very different structural composition of the axoneme between these two. Okay. And that also signifies in their function. This is another variety of cilium that the sensory system makes in Drosophila. And these are the olfactory cilia. And unlike the chorotonal cilium, which is a single long structure, you see that it has this highly branched outer segment, okay, which grows from a little base, which has this basal body and this connecting cilium, which is like a primary cilium. But then it has this huge, branched diversity at the outer segment. So this caught our attention from very beginning, and we asked this question that how does such a diverse structure form from base up, and how does it function? So today I'll tell you how does it function. Now this is the summary of our entire work for last 26 years in one slide. So for this 26 years, I've been fixated with this, to understand the function of this particular molecule called motor protein, kinesin 2 motor protein. And I wanted to understand how does it bind its cargo and transport the cargo. And the reason this makes sense to me, as well as it will make sense to you soon enough, is that kinesin binds to several different kinds of proteins, as you can see here. So we have only displayed about 6 of them, which we found in our lab, but many other such proteins are known. And one of them is orco, which is actually transmitted to the tip of the cilium to make the olfactory cilium smell. Okay. And the other is the acetylcholinesterase and cholinesterase transferase, which goes to the other end of the neuron, which forms a synapse. And what is intriguing to me and as well as many of us is that how does this kinesin 2 motor bind to these 2 different sets of proteins and decide to take them to 2 different destinations inside the same cell. So that's another story which might come to light someday. But today I'll tell you about the olfactory cilia maintenance, and I'll mainly talk about the work done by Varsha and Priya. So this is the olfactory cilium, and this is the place, what you see here in false color heat map is the expression of the olfactory receptor, orco. And this is the place where orco is made, and this is the place where orco is displayed, okay? And see the region in between has no orco, all right? So this is the outline of this whole structure, and what we were interested to understand is that how these receptors which are made at the dendritic end goes up to the outer segment for display. The reason it is critical is because if this receptor is displayed on this region and not in this region, then the fly will not be able to smell, because only this region of the cilium is exposed to the outside world. The rest of the cilium is sealed. So therefore, the cilium also has to have a very intelligent network of sensors which will allow the receptors to position itself at the right position. Okay. So to cut the long story short, we found out that this happens in very small transient steps, which last less than 1 hour during the life cycle of the fly. As the fly emerges from the pupal case, there are these phases of which last less than 1 hour, when majority of the orco receptors are transported into the outer segment. And we also learned that it is specific to orco and not generic to any transmembrane protein. So this is the bacterial channel rhodopsin, which is not present in the fly, and that doesn't follow this rule. So that means the same machinery that is present inside the Drosophila neurons can only recognize the legitimate ciliary proteins and cannot recognize something which it hasn't seen before. And also it is not because the cilia is growing. So this is the growth profile of the cilium, and you see the cilia is not growing, it is static. So which means that this movement are regulated, okay? And we first learned that this movement requires this motor protein kinesin 2, which can bind to these receptors and then move them into the outer segment of the cilia. Now the question that we asked is that, what triggers this episodic orco entry? And the answer is lipid modification. So I'll now tell you how, making some lipid here, changing the lipid composition here can actually trigger the movement through this region of the cilium. It is quite long distance, about 10 microns or even longer. Okay. So before that, I want to remind you once again that the cell signaling is like a domino. Okay. Now, as you will see in this illustration, is that the domino has different rates. And this is the key that I want you to remember, because that will help you to understand the data that I'm going to show you next. So essentially, what the cell has to do now, or rather the cilia has to do now, is to make a clock which will have a variable speed. so that it can trigger different sets of actions at different points. So it has to start some action and stop that action and start some other action and stop that action again. Because during that movement I showed you, there is a start and there is a stop and then there is a delay. So how do you code the start, stop, and delay in this signaling system inside the cilium so that you can achieve this whole intelligent regulation of protein movement without actually investing any neuron in that process? So this, the genesis of this experiment started with this observation, which was made by Priya a long time back, that this lipid, which is called PI4-5 bisphosphate, and this receptor, which is orco connected to GFP, they seem to have complete dislike for each other, and you see a total partitioning, like an India-Pakistan divide. Okay. And so when we observed this, we asked that why so? Why is that phospholipid and the receptor are so antagonistic to each other? And we hypothesized that maybe this phospholipid is the key to regulate the entry of orco, because if there are too much of this phospholipid, then that will prevent the orco to enter inside the cilium. And so Priya looked into the level of this phospholipid, the PI4-5 bisphosphate, and she found that the level of this phospholipid decreases before the level of the orco increases, and there was a very tight phase relationship between the orco entry and the loss of PI4-5 bisphosphate. So therefore, that strengthened our hypothesis that there must be a causal relationship. So to establish that causal relationship, we looked at many different enzymes. Yeah.
Little bit of it'd be great to get a background on how did you decide to look at FIT2 the first?
That's the graduate students' accidents. Okay. So we have been looking at, jokes apart. So Priya has systematically looked at it in many different ways. The first experiment we did was we looked at the membrane fluidity, because membrane fluidity is the main driver of lipid movement and any lipid soluble protein movement inside the cell. But if the membrane is really tight like a butter, you can't move anything. So therefore, we thought that during that phases when the orco moves into the cilium, the membrane must be highly fluid. And we did find that. So Priya did the leveling with body-ceramides, free lipid, and shown that there is a 15-minute cycle in which the ceramide is totally recycled out of the cilium. And she found that those lipid recycling is accelerated during this phase when orco enters, slightly before the orco entry phase. So that was the trigger. Then we found that cholesterol plays a very important role. And as you know, cholesterol is also a very critical factor for membrane fluidity. So then we started asking that what can trigger a membrane fluidity? And that's the answer I'm going to give you. Okay. So we looked at many different enzymes which can modify a phospholipid. And we finally turned out that activity of this phospholipase C, which actually decapitates this lipid, is the key factor that drives the orco entry. So as you see, we can have a dose-independent increase of the phospholipid level by inhibiting the PLC an hour before, and you see the loss of orco that happens an hour after. So again, we found the same, 1 hour phase locking phenomena by monitoring the activity of the PLC. So the summary of that whole thing, and after that, a lot of other things, is the following. So what we found that the loss of phospholipid by the activation of PLC is the trigger that causes the orco to entry, and inhibition of the PLC through phosphoprotein kinase A and adenylate cyclase, which activates protein kinase A, is the trigger that stops the orco entry. So that is the start and stopwatch for orco entry, right? And cholesterol is also important. We still don't know how. So therefore, the PLC activity in the outer part of the cilium will trigger the movement of orcovesicles to fuse at the base of the cilium. And then, somehow the orco will bind to the motor protein, and the motor will push it through the dense, membrane-bound space, a very tight space along the ciliary axon. Okay, so good. So now the question is, what regulates these episodic PLC changes, activity changes, right? So how do you create a PLC2? How do you make PLC2 activate and then inactivate by itself? And that too in this outer segment of the cilium. Now, again, we found that there was the old report, which was made by Geti Hassan and Pinky Kine long time back, that a G protein linked activity, particularly G alpha Q and adenylate cyclase, are somehow involved in Drosophila olfactory activation. And they showed that if you knock down this G alpha Q, then the olfactory activation is reduced. And if you knock down adenylate cyclase, the olfactory activation is increased. So, but they did not give any explanation. And so far, there was no clue that orco could be a G protein. In fact, whatever we understood about the structure of orco is that it is not a G protein. It cannot be a G protein because it has an inverse topology, mirror inverse topology than a G protein. So then how does orco and G protein connect each other and make these things move inside the cilium? So that was the question which Varsha started, and after a lot of other experiments covering all the usual suspects, it turned out that if you reduce G alpha Q selectively in the olfactory neurons at the adult stage, then that will reduce the orco level. And not only that, it will reduce the orco level, it will also increase the phospho-2 levels. So there was the same, complementarity, inverse complementarity maintained even with G alpha Q. So that fit the model quite well, that G alpha Q is involved. And so, and with all other data, we now, came to this hypothesis that perhaps, somehow the odor stimulation, and this is built on the understanding of mouse olfaction, that, it will accelerate the, G protein activity, and that will activate the PLC. And at the same time, another G alpha, which is G alpha S, which will activate the adenylate cyclase. And that will then, somehow activate protein kinase A, and that will then inhibit the PLC. Now, in this process, when the PLC is active, it will, activate. It will make IP3, which will release calcium at the base of the cilium, and that will trigger the movement of motor because kinesin 2 is known to be activated by a calcium-dependent kinase called CAM kinase. So this fits the model nicely, and also there was a data to support this model, which showed that within 2 minutes of 2 seconds of odor stimulation, the PLC is activated in the mouse olfactory cilium. But how does it happen in Drosophila? So does in Drosophila the odor plays the same role in triggering this clock? So Priya tested that hypothesis once again, and by giving extra odor shock as the fly emerges with high concentration of odor, and she found that indeed if you give a high concentration of odor prematurely, then you can actually advance the orco entry phase by 1 hour. All right. So that proves that odor is indeed involved in this process. So then, the Varsha showed that yes, indeed, if you knock down orco, then the orco level is down. Obviously, it should be, but it also increases the PEEP2 levels. So therefore, the orco activity is also required for orco movement inside the cilium. So with these, we finally we still don't know how orco activates the GPCR. But nevertheless, it helped us to build this model of 4 clocks, okay? So which we call GPCR clocks. So for this simplicity, I have divided them into multiple colors. So the green clocks trigger the entries. When it starts, it triggers the entry of the orco. The red clocks, thin red clocks, trigger the stop of the orco entry. So when those clocks are started, the orco entry is stopped. And the orange clock is the delay clock, which will then decide when to, de-inhibit the red clocks, stop the red clock and start the green clock again. Okay. Okay. So this is how it should work, in my opinion. So initially, the cilia is in equilibrium, where these 2 lipids, the PI4 phosphate and PI4 5 phosphate, are in equilibrium. Okay, and this is maintained by these two enzymes called skittled and INPP5. And this has been also tested by Varsha using the genetic experiment. Now, when an order is given, and as the fly emerges from the pupal case, the antenna is free from the pupal membrane, so antenna can smell the food, and then the process starts. And within an hour, this signaling will activate phospholipase C, and that will start the orco entry, trigger the orco entry through kinesin 2. At the same time, at a slower rate, the beta gamma or the alpha S will activate adenylate cyclase, and that will then activate pKa, and which will stop the clock. Yeah? And then the orco movement will stop. So good. So now, how do you restart this clock? Now, up to this much we have the experimental data, so I can be confident about this model. And this is my extrapolation. So now cyclic AMP also activates a phosphatase, PP2A, right? Which will dephosphorylate pKa and phospholipase C. And as a result, phospholipase C will become activable again. And therefore, the antenna will be again receptive to the odor stimulus. And because the antenna is again receptive to odor stimulus, the green clock will start. And then the whole process will repeat itself. Now, will this process continue ad infinitum? No. At some point, the entire membrane of the outer olfactory cilium will be saturated. There will be no physical space left for any further receptor entry. And at that point, the orco entry will stop. And it does indeed stops after the second phase. And that is because you have maximum saturated level of orco. And at that stage, the orco turnover is more than 10 hours. So that's what we found using the trap assay. So that's the summary of the work. And thanks to Priya and Varsha, we now have a good clarity of how this clock works. So with these, I also end my presentation. I just want to leave you with these 3 questions. So how does all this basic fundamental understanding, which looks very nice, helps us to understand a disease system? And will this allow us to cure any disease process for this purpose? I really don't know the answer. And that is what will be my quest in the next decade or so, if I can manage it. But what I believe is that what we understood is that we can pharmacologically control the entry of a receptor into the cilium, entry and exit of a receptor into a cilium. And therefore, in diseases which involves a receptor abnormality where the receptor levels are different, perhaps we should be able to control it by giving this kind of an external trigger, which can be given very easily by incubating the olfactory epithelium externally using these chemicals. So that's my hope. Also, since olfactory epithelium is exposed, or many other such ciliary systems are exposed to the outer world, so incubating the cilium with a particular inhibitor or agonist is rather easy, other than inhibiting a neuron inside the brain or deep inside the tissue. So I hope that these little understandings will ultimately help us to develop some therapeutic solutions in the long term. With this, I end my talk, and I thank all these wonderful people who has worked with me, and now they are no more. Thank you. There I go, except this sitting here.
Not no more there. No more here. So just to make sure. The short sentences. Okay, I see emotional teary eyes. Thank you, Krishanu, for this lovely, very complex story of the cilia, and such a lot to even more do as you have outlined. So thank you. Questions, Shubha?
Yes.
So I continue to be terrified. I mean, if any non-biologically terrified, we are also terrified. But, our lives hang on such a complex mechanism, right?
And it still works.
And it's still,
Yes.
is there an evolution? I mean, we kind of have a sense of how things became complex, not why they're complex. that nature could throw away and if that's working, it keeps adding on patches.
Right,
If something doesn't work right, one more molecule.
right.
But is there a simple thing? Was it always this?
Well, for the unicellular organism, the flagella or cilia function is more simpler in that sense.
Can we understand something from there?
That's what people are trying. So many of these messages that I gave you has derived from the chlamydomonas system. And that has helped a lot to open up the field. But then the other systems like Hedgehog signaling and so on cannot be understood in those unicellular organisms. So for that, we need to delve into the multicellular system and the brain functions. From schizophrenia to the eating disorders, everything seems to converge. In fact, you saw this mouse brain map, right? Where they are now putting together the 1 nanometer resolution images of the whole brain. And they found that there are these hippocampal neurons grow a cilium, which also expresses serotonin receptors, which make synapses with the axon. And the signaling through those serotonin receptors have a different outcome than the signaling through the dendritic serotonin receptor. So it's really complex. So the short answer is, more exciting time or terrifying time is ahead. Yeah. Uncle?
In context, because the entire machinery to it also is responding, that is the PLC, etc. It's also there on the segment. So has somebody tried to put work on the segment and see if it is
Doesn't function. So that is true for all GPCRs. In fact, rhodopsins and orco and many other GPCRs, they don't function on the platform. They have to go to the cell. Rhodopsin, for example, unless it is in the rod outer segment, it will not sense light. And so is orco.
So is there some structure? Because
Yes, there is. So what happens is it's the lipid ordering that is the critical function, because the receptors in the transmembrane proteins, the lipid environment in the transmembrane region plays a very distinct role in controlling the receptor activity. So rhodopsins and all other GPCRs, they have to bind to free cholesterol. And only when the free cholesterol binds to them at a particular groove in the transmembrane helix, the receptors become active, or activable rather. So until then, the receptor is not activated. So similarly, the phospholipids play a very critical role in other receptors like TRP granuloid. It can only function with PI4 phosphate and not with 4,5. So depending on that, the lipid environment, the ceramides have a different role in that process. So the lipid metabolism, again, local lipid metabolism in the cilia is very, very important for the ciliary function. And that's another whole field by itself now.
Can I ask a question?
Yeah.
How does the apex of the cilia and the base maintain a difference in the phospholipid content? You said this
by
is
distributing the enzymes in different locations. So the tip has one kind of enzymes, the base has another kind of enzymes.
so there is a gradient, right?
It's not a gradient.
It's
a very,
collection.
very,
It's a very
very structured
structured.
positioning of these proteins, enzymes, that actually shapes the function of the cilia.
But this should be energy consuming. Otherwise, the free energy should tend to
Life doesn't care about energy economy.
No, I think it should, right? In some sense. Right?
So life has evolved, not because of energy economy. Life has evolved because of energy
But it
availability.
cares about energy economy. It may not have evolved by that.
Life in and of itself is an entropy reversal unit.
No, that's okay. But at some point, you are doing work
Seriously speaking, the life cannot care about energy economy is because the energy supply, because these are all molecular machines, and therefore they will act when they have the purpose and when they have the chemical environment to act. Now, how do you coordinate the chemical environment of the whole diverse system by itself? It's not feasible, right? I mean, they're not linked with each other. So therefore, you cannot build an energy economy in a multi-organ or multi-unit system. either it's an ecosystem or it's a multicellular organism, whatever you look at, at least in case of a multicellular organism, you have some control. You can control the input output and control the energy supply. But other than that, nothing. And
Locally speaking, maybe there is not necessary of an energy balance, but overall it has to be. Locally
but
speaking,
who
even
will-
I'm
no, but who will coordinate it?
yeah, that
So
was
you
my
cannot
question.
coordinate that.
That was my question. You said enzymes are positioned. And
Huh.
they
So enzymes are positioned. Again, enzymes are positioned in different system in a different way.
Okay.
And that again is an evolution that happened, and I don't really know how.
Okay. Any other questions to Krishanu? If there are none, let's have a big round of applause. Thank you, Krishanu. Doing this on a Saturday, and thank you all for coming. There is some tea and coffee outside. The rest canteen will have to walk out and come back on the outside table. So thank you very much and please join. And I would like to thank my student Ayush who did the AV. Thank you.
Thank you, Krishanu. All the best.
Thank you.
All the best. Mahendra. Thank you, Mahendra.